Hi friends, so now I'm back with my next video, and in this video we are going to talk about translation in eukaryotes. So in my previous video we have talked about the prokaryotes, how it process, and then the recognition sequence like the Schindel-Gano sequence. And in this video we are going to talk about the differences between the eukaryotes and prokaryotes and the factors which are required particularly for eukaryotes. So please make sure to like, share and subscribe my channel. So as I have talked, what is translation? It is the process of protein synthesis in which the mRNA get translated to the protein, which is a sequence of amino acids. And then the protein synthesis, it usually proceeds from N-terminal to the C-terminal, and the mRNA is read in the 5 to 3 direction. So if you remember, I have talked about the different direction like the 3 to 5 strand of the DNA act as a template strand for the mRNA synthesis, and the mRNA that is produced is in a 5 to 3 direction. So the ribosome read the mRNA in a 5 to 3 direction. So here you can see the DNA. So one of the strand here will be in 3 to 5, may this one, and then the RNA which is produced is read from 5 prime to the 3 prime, right? And ribosome always bind to the 5 prime site and then continues until the 3 prime site. And then the translation, where does it take place? It takes place in the cytoplasm of the cell. And as I have told in my previous video, that in eukaryotes the mRNA, which once it's formed, it get further modification like splicing and capping, polyadenylation in the nucleus, and then it get transported to the cytoplasm where the ribosomes are present, and then protein synthesis takes place. So what are the components required for translation? So as we have talked in the previous video as well, the mRNA, which is made in the nucleus and then transported to the cytoplasm, tRNA molecule that transfers the amino acids to the ribosome and also decodes the information in the mRNA which is present. And then obviously ribosomes, which are the manufacture for the proteins. And then there are different enzymes required for the attachment of tRNA, for the formation of peptide bonds between different amino acids, and other factors like the initiation factor, elongation factor and termination factors, which are different for the eukaryotic translation. So we have talked about the ribosomes, which are required in the prokaryotes, which is consist of 30s subunit and 50s subunit. But in case of eukaryotes, it's a bit different. In eukaryotes, the larger subunit is 60s subunit and the smaller subunit is 40s subunit, which contain different ribosomal RNA and different proteins as well. So now that the ribosomes are different, so we can talk about the binding sites, which we have also discussed. The binding sites are the same, the E site, the P site and A site. A site is the site where it binds to the incoming tRNA, and then the peptidal site in which the growing polypeptide is attached, and then the E site where the uncharged tRNA, the tRNA which is not bound to any amino acids, is present, and then it departs from this site. It's the same as in prokaryotes. So I have also already talked about the charging of tRNA, like how does it occur? By the action of the enzyme amino acyl tRNA synthetase, which binds to the amino acids in the presence of ATP, and then the tRNA, which contains the anticodon sequence at the 3N, it binds, comes, and then the amino acids with the tRNA is released, which is called the charged tRNA, and then the what is released is AMP. So this process of charging of tRNA and different sites on the ribosome, it's the same in prokaryotes and eukaryotes. So now we have the charged tRNA, which is required for the initiation. So initiation in case of eukaryotes, it's a bit different as compared to prokaryotes. So in eukaryotes, the initiating amino acid is methionine instead of the N-formyl methionine. So the codon is same AUG, which is recognized, but the amino acid which is required for initiation is methionine instead of the N-formyl methionine. And as you remember from the prokaryotic translation, to initiate the translation, the 30S subunit, it recognizes a sequence in the prokaryotic mRNA which is called the Shine-Dalgarno sequence. But in case of eukaryotic mRNA, there is no Shine-Dalgarno sequence is present. So what happens is the 40S ribosomal subunit, it binds to the 5' end of the mRNA and moves until it finds a AUG initiation codon, which is present inside a Kozak sequence, which I will talk later. So this process of binding and then moving and recognizing the start codon is called the scanning. And then there are different, like 9 different eukaryotic initiation factors in eukaryotic translation as compared to the 3 initiation factors which were present in prokaryotes. So as I have talked about, the initiation codon is usually like AUG. It is contained in a short sequence which is called the Kozak sequence. So here you can see ACC AUG. This is what is recognized by the 40S subunit within this sequence, because you can have multiple AUGs present, but only when AUG is present in this sequence it is recognized as a start codon. So here is the initiation process. So there are different steps which are involved, the initiation factor as well. So first is the formation of this pre-initiation complex. So how does it form? The EIF, elongation factor 1 and 1A, they are important for the scanning, like the movement of the subunit along the mRNA. And then EIF2, which binds to the tRNA, the first tRNA, methionine, which is binds to the 40S subunit. So here you can see the 40S subunit, the tRNA methionine, and then different elongation factor like 1 and elongation factor 2, like which is catalyzes the exchange of GTP to DDP. And then AIF3, it also prevents to the 40S subunit and prevents the binding of the 60S subunit. So until it binds to the mRNA, the EIF3 is required to prevent the binding. So once this complex is formed, it binds to the 5' end of the mRNA and starts the scanning with the help of EIF1 and 1. So once it moves, it finds the start codon AUG, right? And then it all different initiation factors get released, and then this EIF5, it stimulates the binding of the 60S ribosomal subunit to the small subunit. So once this complex is formed, the initiation has done, and what starts is the elongation. So elongation is almost the same as the prokaryotic elongation, except the factors. So instead of EFTU and EFTS, which are present in prokaryotes, in case of eukaryotes there are EIF1A and EIF1B. So we have the first tRNA, which is methionine, present at the P site, and then another amino acid comes to bind at the A site, another tRNA that binds to the A site, and with the help of transcription factor 1A, and then this methionine get transferred to this tRNA, and what left is the uncharged tRNA. And with the help of the EF2 or EFG in case of prokaryotes, it get translocated, like the uncharged tRNA get translocated to the E site, and then this tRNA with 2 amino acid get translocated to the P site, and then there is space for the another tRNA to comes, and the process continues until we find the stop codon. So the termination is exactly similar to the prokaryotic termination. So once it recognizes a stop codon UAG, UAA or UGA, it arrives at the A site. It is recognized by the release factor 1 with the help of ERF3, the release factor 3, and it terminates translation. And upon termination, the ribosomal subunit is disassembled, the polypeptide is released, and then this mRNA is also released. So this marks the termination of the eukaryotic translation. So I hope it's clear the difference between the prokaryotic translation and eukaryotic translation, different factors required, and what is the difference between the Shine-Dalgarno sequence, which is the sequence important for the initiation of prokaryotic translation, and the Kozak sequence for eukaryotic translation. So I hope you like this video. If you have any doubts, please let me know in the comment section, and please make sure to like, share and subscribe my channel. Thank you.